supposed to be blood and cardiorespiratory system. We have already addressed the respiratory system. We have addressed the cardiovascular system, that is the blood and the blood vessels, and the heart and the blood vessels. And we noted that all these systems are interrelated. One of the common factors is blood. Blood has to supply the lungs for oxygenation. The heart has to pump blood to the lungs for oxygenation. And therefore, we are going to look at the blood, and in relation to this, we are going to see where some of the formed elements of blood originate from. The formed elements of blood are the cells of blood. So we are going to see where they originate from and how they are activated to form the immune system. So we'll be looking at the structure of the lymphoid organs, blood, and then structure of the lymphoid organs. But first, I'd like us to look at our blood. And looking at blood, we are familiar with the blood right from our high school. We know that it's made up of different cell types. So we are going to look at the histology of these cell types. The immunologists will tell you about how they get activated for immune system. The physiologist will tell you its role, physiological role in the immune system. The biochemists will tell you about the composition of blood in terms of biochemical nature of the blood, and even within the cells. Like hemoglobin, they will show you the structure of the hemoglobin and how it is able to bind to oxygen. But for anatomy, we are looking at basically the structure of the cells. The structure of the cells, and you might mention here and there the functions of these cells in relation to the structure. So let's look first at the functions of blood. Why do we need blood? Hence, we look at the functions of blood, and we noted that it's very important in the transport of gases. Both oxygen, primarily transport of oxygen to the lungs and to the tissues, and also transport of carbon dioxide in the dissolved form. Normally, carbon dioxide is transported either by carbonic acid or bicarbonate. So that's important in noting that this transport of gases, carbon dioxide from the tissues, oxygen to the tissues. Then we have metabolites. The cells metabolize substances. Some of those metabolic products are then transported by blood. For example, urea, which has been metabolized, say, in the liver, will be transported by blood to the kidney where it's removed. Nutrients, once they get absorbed in the gastrointestinal tract, will also be transported by blood to the tissues and hence to the cells. And also, of course, hormones. Hormones are produced by endocrine glands, which do not have ducts, but the products, which are the hormones, are transported either bound in proteins within the blood, and therefore they're transported to their target organs. Then we have distribution of heat. Heat is normally generated where you have a high metabolic activity. We have heat being generated in the liver, primarily. It's being generated in the muscles. For example, during exercise, there's a lot of heat being generated. This heat is then distributed throughout the body by blood, so that you have what we refer to as the core normal body temperature. The core normal body temperature. This is because of the heat being transported from the origin where it is being generated and being distributed throughout the body. Regulation of pH, we have carbon dioxide being removed, for example, in the form of carbon dioxide, but being removed as carbonic acid. Part of it is carbonic acid, which is then converted to carbon dioxide and being removed. During that conversion, some of the hydrogen ions will be remaining in the blood. And therefore, that can be regulated through the both respiratory system, like during breathing, and also we have excretion of hydrogen during urine formation, where we have the hydrogen is being excreted in the distal convoluted tubules of the kidney, and therefore there's acidification of urine. In the process, then we are removing part of the hydrogen from the body to regulate the pH of blood. As a result of this, there's also regulation of the interstitial fluid, or part of the intercellular fluid. Then we have leukocytes, the white blood cells, participating in the immune system, white blood cells participating in the immune system. So again, we are going to look at the structure of the white blood cells, and we'll look at the organs in which some of this activation takes place. So we're going to look at the structure and the structure of those organs, the cells themselves, and then the organs. Then, of course, if there's any injury, there's clotting of blood. So we have clotting factors within the blood, and that is very important, the platelets and other proteins that are necessary for coagulation of blood. So we're going to look at this also in relation to the platelets, the structure of the platelets, and also where they are formed from and how they are formed. So in relation to this, blood, when you centrifuge blood normally, we have what you call a hematocrit. A hematocrit is where you have whole blood being centrifuged. There's no clotting. When you clot blood, you have serum, but in blood which has not clotted and is centrifuged because an anticoagulant has been added, then you have normally 55% of the volume will be plasma and 45% will be formed elements. Now we'll just mention the composition of plasma, but generally, biochemists will go into the details of the various proteins that we have within the plasma. Formed elements are the cells and fragments within the blood. Cells and fragments within blood. These are blood cells and also fragments. In this case, we are talking about the platelet, which are not cells, but are fragments of megakaryocytes. So the formed elements have erythrocytes, leukocytes, which are white blood cells, platelets, which are not cells, but are fragments. Normally in a hematocrit, the red blood cells will settle right at the bottom. Then we have the plasma, and we have what we refer to as a buffer coat. A buffer coat is a very thin layer of white blood cells. Very thin layer of white blood cells. Of the leukocytes, we'll be looking at their details, but they're usually referred to as granulocytes. These are the ones which have granules in them. I'm told I'm not audible enough. Let me change my microphone. Thank you for that information. I hope I'm audible now. Am I audible, class rep? Gillian, are you able to hear me?
Yes, we can hear you.
Fine. Let me continue. So we'll be looking at the leukocytes, which are white blood cells, normally are classified as granulocytes and agranulocytes. Granulocytes are those which have granules in them. They have granules in them. And then agranulocytes are those who do not have granules. Now, when you talk about granules, we're talking about specific granules. Specific granules. Of the granulocytes, normally neutrophils are the majority, followed by eosinophils. Basophils are the least. Basophils are the least. So we'll be looking at them in terms of their composition. And also we have granulocytes, which are the monocytes and leukocytes. Now, granulocytes, when you have A before a cell, a prefix A, it is negating what follows it. So granulocytes do not have specific granules, and we'll be looking at them in details in terms of their structure and why they are able to perform their function. Now, plasma is usually, you refer to it as straw-colored, not completely clear because of what it contains. And part of what it contains will be a red blood cell breakdown of hemoglobin. The hemoglobin breakdown can usually, you can have biliverdin, bilirubin. So all those could be within the plasma, and therefore that gives it a straw-colored and not clear. 90% to about 92 is mainly water, and then 7 to 8% are proteins. And of the proteins, albumin is a major protein that we have in blood, and albumin is synthesized in the liver. We also have globulins, and these globulins will include the immunoglobulins, which are very important in the antibody reaction. And then we have transport proteins, which are dissolved in plasma. The transport proteins are very important in transport of, say, hormones and other substances. And then fibrinogen, which is very important in the clotting. So those are the proteins. We also have electrolytes in blood, that is sodium ions, potassium, calcium, the chloride and bicarbonate ions. All these are electrolytes that we have in blood, and this is the main composition of plasma. And apart from this, the nutrients that are being transported from the digestive tract to the tissues, from the digestive tract to the tissues. And of course, the metabolites, after metabolism by the tissues and waste products, these again are being removed, and therefore they are removed in plasma. They are part of the plasma that we noted. I talked about urea a short while ago, but also bilirubin, biliverdin. This is the breakdown of hemoglobin. When red blood cells are destroyed in the spleen, then part of the product is bilirubin, which is then transported in plasma before it's removed, either in urine or in fecal material. So this is what we have as part of the components of the plasma. Apart from this, of course, we have dissolved gases, carbon dioxide and oxygen. Yeah, oxygen, which is dissolved usually not necessarily being transported by the hemoglobin, but being dissolved in a physical solution form. And then hormones and enzymes, all these are within the plasma. So when you look at plasma, we have to have this in mind, that we have all these structures. And some of these elements also are very important in nourishing the cells within the blood. So the cells within the blood are active and they need nutrients. Some of these nutrients are also important for those particular cells. Now, for the formed elements of blood, we use the word formed elements because these are not originally in the liquid form. These ones are cells which are formed elsewhere but are transported within the blood. They enter circulation, they can leave circulation, and then they're transported throughout the body. So red blood cells are the major formed elements of blood. These are the erythrocytes. We'll see where they are formed, most of them in the bone marrow and especially red bone marrow. And all these other cells are mainly formed in the bone marrow. But we will see what happens during embryological development. Then we have granulocytes, like I had mentioned, the neutrophils, basophils, and eosinophils. And then we have agranulocytes, which are the lymphocytes and monocytes. And then thrombocytes, the platelets. Thrombocytes are the cells which form platelets. Platelets are actually fragments of the pseudopodia of the thrombocytes. And thrombocytes are resident in the bone marrow. So they are resident in the bone marrow, but their fragments now pinch off, enter circulation as platelets, and are able to move to all parts of the body, helping in blood clotting. And you can just note that platelets have a lifespan of 10 days, 0 to 10 days. So it's a very short lifespan. So a lot of it is being generated every day, every minute. As a general statement, we would say all formed elements originate from bone marrow. They originate from the bone marrow. And within the bone marrow, we have these hematopoietic stem cells. Hematopoietic, that means they produce their stem cells. That means they multiply, differentiate to form other cells, specifically hematopoietic. And of these stem cells, they are usually divided into two. We have the myeloid series. We are going to see which cells originate from the myeloid series and the lymphoid series. So in general, we can say that apart from the lymphocytes, all the other formed elements originate from the myeloid series. So they originate from a common cell that is myeloblast after the stem cells have been formed. Normally, we have what we call burst-forming cells. These burst-forming cells then form colony-forming cells. When they multiply and differentiate, they form colony-forming cells. Those colony-forming cells now specify within the myeloid series or the lymphoid series. So in general, the bone marrow in the adult as universal hematopoietic organ. All the formed elements come from the bone marrow in the adult. But in the fetus, during development, we are seeing a different scenario. Okay, we're seeing a different scenario. Now, of the formed elements, we said neutrophils are the most common ones. When you look at any of the blood smear, what you would see are neutrophils commonly. You see lymphocytes also quite a bit, but neutrophils of the formed elements of the granulocytes are the most common ones. And these ones here have a general lifespan of 8 days. So because they are very important as surveillance, they are usually found in the body. They are first line of defense. When there's an injury anywhere, neutrophils are among the first cells that arrive in that area to fight the injury, to fight the infection, to remove the debris. And then within the loose connective tissue, we have some of these cells in circulation, moving within the connective tissue. So they are within the connective tissue as surveillance cells, apart from the other cells. Now, neutrophils, we recognize them are multilobed. having a multi-lobe nucleus. This multi-lobe nucleus, normally 2 to 5 lobes of the nucleus, are connected by some thin chromatin strands. Thin chromatin strands. We'll just see a picture of this. Multi-lobe, 2 to even 8 lobes of the nucleus. But all these are connected together by chromatin strands. Usually, on one of the lobes of the nucleus, we have a club-like extension that we refer to as a bar body. B-A-R, bar body. The bar body actually contains condensed chromatin, which is sex chromatin. And in most cases, when you look at the blood smear and we're able to identify bar body, in most of the cases, that blood will be from a female, because the bar body is linked sex chromatin. Then we have organelles within the neutrophil. Now we'll be looking at the function of the neutrophil. And then we want to generate, having known what the organelles do, what the organelles do, you should be able now to relate those organelles to the cell. We'll just go to the structure of the neutrophil first, and then we'll be able to look the function of the neutrophil, and then we're able to look at the organelles later. Now, staining, both acidic and basic dyes take up stain, and that's why they are called neutrophils. They stain for both acidic and basic dyes. So usually in a blood smear with the H&E, there will be pale staining, normally pale staining, because this both acidic and basic, they have affinity for both basic and acidic dyes. This affinity is because of the composition of each cytoplasm. Within the cytoplasm, we have granules, and that's why they are called specific granules. That's why they are called granulocytes. Granulocytes. Some of the granules include lysozymes, phagocytin, alkaline phosphatase. These are specific granules. But we have non-specific granules, like lysosomes. Lysosomes are in granular form. Those are non-specific. These are also, the non-specific are also referred to as azurophilic granules, because they take up stain with azure stains, dichromate stains. Now, the activity of the neutrophil, usually they produce cytokines and other, if there's any injury, there's production of cytokine and other inflammatory factors, and this influences inflammation. So when there is an injury, the cell produces products from the cell. This refers sometimes as cytokines or chemokines. These ones, therefore, the neutrophil is able to modulate the activity of the neighboring cells, so that these particular destroyed cells or the bacteria that are adjacent to the cells can be able to be destroyed. In a sense, they also regulate the activity of the macrophages, regulation of activity of macrophages. Macrophages are phagocytic, just like neutrophils are also phagocytic. And neutrophils also play a role in innate immunity, and that is for memory and cancer cells, construction of cancer cells. So we need to, physiologists will tell you, immunologists will tell you. But of importance for us is to look at this neutrophil. Look at this neutrophil in terms of, this is a neutrophil amongst erythrocytes, and you can see 1, 2, 3, 4 lobes. There are 4 lobes, and these lobes are connected by a thin strand of chromatin material. Usually, there might be 2 to 4 lobes, 2 to 4 lobes, or even up to 8. The number of lobes increase with age as the neutrophil becomes more mature. Within the cytoplasm, we'll be having granules which are pale staining, not strong staining, but pale staining. We are going to compare this with those of eosinophils. In one of the lobes, you see a club-shaped structure. This club-shaped structure, which is an extension of the lobe, the nuclear lobe, is part of the sex chromatin. Sex chromatin is what we are calling the Barr body. Now, the functions of neutrophil are, one, something you are very familiar with, is phagocytosis. So if we are going to phagocytize a substance, what would be the main organelles within the cytoplasm? We expect, of course, the lysosomes that should be able to destroy the phagocytized material. We expect the ribosomes to be able to synthesize the proteins. We expect rough endoplasmic reticulum synthesizing the proteins that are necessary for the lytic enzymes that are in the lysosomes that are necessary for the destruction of the bacteria. We expect the Golgi apparatus. Golgi apparatus is very important in the formation of the lysosomes and the conjugation of some of the proteins. So those are the major organelles that we have. Apart from this, in phagocytosis, there is movement of the cell membrane. That movement of the cell membrane is achieved by microtubule, polymerization of the microtubules, forming a pseudopodia, which come to surround the bacteria. So microtubules are very important in intracellular transport. During that process, we also need energy. So we have mitochondria providing the energy. You can see a lot of membrane-bound structures. A lot of these membrane-bound structures will need some smooth endoplasmic reticulum for the formation of intracellular membranes, for the membrane-bound structures, like now the phagosome, eventually becoming larger. This is one of the modes of destruction, that is phagocytosis. The other mode is release of granules, that is degranulation. The granules are released, and those granules, once they have been released, among the granules that are released is lactoferrin. Lactoferrin granule released will be able to then destroy the cell membrane of the bacteria, and therefore the bacteria is killed. Another process is called netosis, and in netosis we have the elastase. The DNA is released to the outside. We have the elastase, which directly kill the pathogen, but we also have myeloperoxidase proteins which trap the bacteria. So this helps in trapping the bacteria. I think physiologists will go into this mechanism with mitosis. So that is the function of the neutrophil. So in a blood smear, we are able to recognize the neutrophil. Notice multilobed nucleus, small strand of chromatin material that connect them. This one has up to 5 lobes. This one has 4 lobes. This one has about 3 lobes. And you can note also the Barr body. So these are the neutrophils you're able to recognize here. 1, 2, 5 neutrophils in this blood smear. This other one is an eosinophil. Notice it has only bisegmented nucleus connected by chromatin material, bisegmented nucleus, and very dense granules, very, very dense, large granules, as compared to the neutrophil. We'll be looking at the others later on. This is just at a high magnification, one of those you are seeing, to demonstrate the Barr body. The Barr body here. So these are a blood smear. Now, the eosinophil, the word eosin is usually acidic. So it has an affinity for acidic dyes. So that's why it's called eosinophil. The granules have affinity for acidic dyes, and the eosinophil is just almost same size like a neutrophil. It has a bisegmented nucleus connected by strands, lobule, bisegmented nucleus, lobule, connected by a thin strand of chromatin. And within the cytoplasm, like I've said, we have very large, dense granules. Very large, dense granules. Compare the size of the eosinophil with the red blood cell. And you can see the eosinophil is far larger than the red blood cell. And in general, all the formed elements, we normally have the granulocytes are the largest because they have a lot of granules in them. We'll be looking at the relationship between the size of the neutrophil and the lymphocyte and also the erythrocyte. But granulocytes are normally relatively larger in size. So eosinophils constitute 1 to 4% of the cells in circulation. 1 to 4% of cells in circulation with a lifespan of 8 to 12 days. Lifespan of 8 to 12 days. After that, it will move from the body. Then we have usually bilobed or bisegmented. Means it's two lobes which are just connected by, not necessarily connected by a segment, a chromatin strand, but they look like C-shaped, V-shaped. That's what we have of the nuclei. And then the organelles we are going to look again to determine what organelles do we expect to see in an eosinophil based on their function. Let's talk about the function, then we talk about the organelles. So in general, the activity of eosinophil is inhibited by adrenocorticotropic hormone and cortisol. Cortisol, these are steroid hormones. Steroid hormone, cortisol is a steroid hormone produced by the adrenal gland. So this suppresses the activity of the eosinophil. Now, eosinophil is one of the cells normally in loose connective tissue acting for surveillance. What does it do? What is the function of eosinophil? The eosinophil contain granules. The dense granules we're talking about are very large granules. These ones are histaminase. Histaminase. We notice histaminase is an enzyme. Enzyme that degrades histamine. Histamine. During allergic reaction, normally histamine is produced. So when histamine is produced, the eosinophils degranulate, release the granules, and the granules neutralize histaminase. They also have heparin, and heparin plays a very important role in coagulation of blood, and the cytokines. The interleukin, for example, as a cytokine, is also within the granules, specific granules of the eosinophil. So in general, we can say eosinophils play a very important role in allergic reactions. So usually during allergic reactions, the eosinophils degranulate, and also parasitic infections. So part of the granules that are released are able to kill parasites that are toxic to the parasites. So that's in parasitic infections, they then have a lot of eosinophil. So if somebody has a lot of internal parasites, there'll be a lot of eosinophilia, a lot of eosinophil in blood. Now, cytokines, which we talked about, promote proliferation and activation of T cells. And remember, T cells are part of the immune cells. Leukotrienes mediate allergic reaction apart from the histamine. And then they also act as antigen-presenting cells by expression of MCH2. They disrupt helminth membrane. They disrupt the helminth membrane. So the helminth membrane is destroyed, and therefore the helminths are destroyed. The worms are destroyed. That's why it's very important, or you have a lot of eosinophils in circulation during allergic and parasitic infections. And then this idea of playing a role in major histocompatibility expression is also very important, and in relation to activation of T cells. So this is the role of eosinophils. And that's why we have a lot of granules, large granules in their cytoplasm. For the basophils are slightly smaller, very few in circulation, 0 to 5 to 1% in circulation. So in a blood smear, we have to look quite a lot to be able to see basophils. They are not very easy to locate. It will be very easy to locate the neutrophils, very easy to locate the lymphocytes, but very difficult to locate the basophils because they are very low percentage in circulation. Usually, nucleus is kidney-shaped, like what we are seeing here. It's a kidney-shaped nucleus, Sometimes the nucleus is almost C-shaped, virtually kidney-shaped with the indentation. And then it has a basophilic cytoplasm. So the cytoplasm stained with basic dyes. That's why they're called basophils. They have affinity for basic dyes. And a very, very short lifespan, 1 to 2 days. Now, what organelles do we expect to see in circulation? Bravin, can you mute your microphone, please? Bravin, mute your microphone. Organelles that we expect to see depending on what are the granules. Do we have any granules in them? Yes, there are granulocytes, so we have granules. The granules are usually very large. They contain histamine, heparin, histamine, heparin, leukotrienes. These are protein. They'll be granules, so they're membrane bound. They're membrane bound. They are packaged by the Golgi. They're produced by ribosomes, so we have a lot. We have, of course, intracellular movement require energy, so we have mitochondria. Intracellular movement require microtubules for moving this both the granules that have been formed. So this is what we have. Now, granules that are released usually play a very important role during inflammatory condition. They play a very important role in inflammatory condition. So basophils in nature are very important in inflammation. Therefore, we have a lot of basophils within loose connective tissue, eosinophils and basophils. So they bind to B cells to stimulate formation of immunoglobulin E. Immunoglobulin E, which is very important in inflammation. It's also an antiparasitic and antivenom. The granules that are being produced are antiparasitic and also antivenom. Therefore, that's very important for inflammation. Now, heparin, very important for blood clotting. Histamine causes vasodilation. So if you have an injury in any place, these tissues are damaged, these leukotrienes are produced, cytokines are produced, this vasodilation to try to get more blood into the region and also to try to dilute the irritating material. In getting more blood, therefore, basophils will be very important in recruiting more blood cells to the region and hence the B lymphocytes coming to the region, and also the neutrophils, which are coming to mob up the destroyed cells and any bacteria that might have damaged the tissue. So basophils play a very important role in the inflammatory reaction. Inflammatory reaction. So in general, granulocytes, one, neutrophils play important role in phagocytosis. They are first line cells to come, first line of defense, cells to come when there's any injury. Basophils will be there also to increase blood flow to the region so that you can have more of the antibodies being produced, being sent to the region, more of the B cells being sent to the region, and also more of the clotting factors being sent to the region, also the platelets being sent to the region to try, for example, to patch up the vessel that has been damaged. So we can say about inflammation. And therefore, when you get injured, there's a swelling. Why is that swelling there? Because of the effect of basophil. The place also becomes warm. After an injury, if you touch the injured area, swelling, you'll note that it's warmer than the other areas because there's more blood coming to the region. There's also a lot of cellular activity generating more heat. Now, we look at the granulocytes. Granulocytes are those cells which do not have granules in their cytoplasm. They have granules, but the granules are not staining. They might be too small to stain, that's one. They might not be picking up any stain with the dyes, the normal dyes, that is a basic and acidic dyes, and that's why they are referred to as agranulocytes. Lymphocytes are agranulocytes. I think those are the ones you are very much familiar with in terms of immunology. Lymphocytes originate from the lymphoid series. They are agranulocytes. They are very large. We are able to look at two different types of lymphocytes in histology. We have either large lymphocytes or small lymphocytes, large or small lymphocytes. We are not able to distinguish the B lymphocytes, and I'll answer the questions, I think, at the end of the session. We're not able to distinguish between the B lymphocytes and T lymphocytes. What you're able to see in a slide is either large or small lymphocytes. Now, of the lymphocytes, we have usually general characteristic will be a very large spheroid nucleus, round nucleus, very large round nucleus, occupying almost the entire cytoplasm, with a very thin peripheral cytoplasm. So this is the nucleus, very dense chromatin, and then here we have a very thin cytoplasm forming a rim on the outer part of the nucleus. So this is a lymphocyte. I told you before to look at the structure. Notice, this is a lymphocyte, this is an erythrocyte. And you can see that the size of the lymphocyte is just slightly larger than that of a lymphocyte, but not as large as that of the granulocytes. But we'll be seeing an exception. Now, we normally have histological types such as the large or small. But you'll be told about B cells, which when they are activated, they become plasma cells producing antibodies. And then T cells, which are either T helper cells, which are activated immune cells, and cytotoxic T cells, which can kill infected cells. These are the T cells, type of T cells, T helper cells, and cytotoxic T cells. And then the natural killer cell, which is slightly larger than the B cells, and these are the cell types, natural killer cells. And these ones here are able to destroy viruses, and also the infected cells of cancer cells. Cells infected with the viruses or cancer cells are able to be destroyed by the natural killer cells, and therefore they are larger normally compared to the other T lymphocytes. But in histology, we're only able to distinguish the large or small size. And then the immunologist will tell you further, classify them further using the word CD, CD8, CD4, CD, whatever. So that one there, let's leave that to the immunologists because of immunostaining. The other granulocyte is the monocyte. Monocyte is the largest cell, very, very large cell, the largest of the formed elements of blood. So these are monocyte nucleus, very large. The cell itself is fairly large. You are not seeing quite the outline. What you're seeing is just the nucleus. And just like the other one is produced in the bone marrow by the myeloid lineage. Now, of coincidence, towards the end of the myeloid lineage, both the cell produces the monocyte is the same cell that produce the neutrophil. The monocyte and neutrophil are produced by a common cell within the myeloid lineage. And when you look at their function later on, you'll be able to appreciate why. So it's the largest cell. It undergoes mitosis, capable of undergoing mitosis, even after differentiation, so that you can produce more of those. Why do we need to produce more? Because these are very, very important cells. A monocyte, when it leaves the circulation and goes to the tissues, becomes a macrophage. And a macrophage is one of the cells of loose connective tissue, cells circulating all over. Whether in the lungs, we talked about the dust cells, to be able to destroy, phagocytize substances. So we talked about the nucleus is intended, folded nucleus, but it's very large. Now, when it leaves circulation, it can either be fixed like a microglia in the brain, is a macrophage, originated from a monocyte. Within the liver, we have cells we call Kupffer cells, which are macrophages, which originated from the monocyte. In the skin, in the epidermis, we have Langerhans cells. Langerhans cells of the skin are fixed macrophages originating from monocyte. Dust cells in the lung, I think we talked about this. Osteoclasts in the bone, chondroclasts in cartilage. All those have origin from the monocyte. So when the monocyte leaves circulation, it becomes a macrophage. Then we have two types of macrophages, the fixed macrophages and the wandering macrophages. The fixed macrophages I've mentioned here. The wandering macrophages are all over. They mainly form what you call dendritic cells in tissues. Wandering macrophages are within the loose connective tissue, acting for surveillance. And these are very important in innate and adaptive immunity, which we talked about. Immunologists will expand on that later on. This is just a monocyte looked at from a different angle. Note is a very large cell. This is a lymphocyte. This one here is a red blood cell. Notice the size of the lymphocyte vis-a-vis the size of a monocyte and the size of an erythrocyte. You will appreciate that the monocyte is the largest. It does not have any granules. However, its role is phagocytosis when it leaves circulation. So when it leaves circulation, it becomes a macrophage. Macrophage is phagocytosis. Neutrophil is phagocytosis. So you can look at them in terms of similarities in the type of organelles that they will be having within the cytoplasm. thrombocytes are platelets. Platelets do not leave the bone marrow. Platelets are very large cells in the bone marrow. They do not leave the bone marrow. They have a lot of processes. Some of these pseudopodia processes, foot-like processes, finger-like processes, project through the sinusoidal capillary openings into the capillary. We know the bone marrow contains sinusoidal capillaries. So they project through the sinusoid into the sinusoidal capillary, and the tips pinch off. They pinch off to form platelets. So the pinching off is just cytoplasmic content. It does not include the nucleus. It does not include the nucleus. So we'll be looking at the platelet to look at their structure. So the thrombocytes produce pseudopodia, and this pseudopodia, like I've said, they project into the sinusoids and pinch off to form platelets. So within the bone marrow, the thrombocytes have what you call megakaryocytes. They're also called megakaryocytes. Mega means large, karyocyte, very large nucleus. Very large nucleus, sometimes up to double the chromatin material, very large nucleus. And this is coming out because of what you call endomitosis. This, or what you can call karyokinesis without cytokinesis. There is the nucleus doubles, chromatin material doubles, but the cell does not divide. Now, the platelets, that was megakaryocyte. Now, the platelets, which are in circulation with a very short lifespan, have no nucleus. They have no nucleus. However, when you look at their structure, we note two areas. We have an outer part of the platelet and an inner dense core. The inner part is dense. The outer part, which is rather light staining, containing something almost similar like the cytoplasm, is made up of what we refer to as the hyalomere. Then the inner part contains usually granules, dense granules that are very important in the process of clotting factors. So this inner part is what we call granulomere. So an outer hyalomere and then a dense inner granulomere. So all this is surrounded by a membrane, a cell membrane, which was part of the membrane of the megakaryocyte. Now, the platelet usually is smooth. It's usually very smooth on the outside. The membrane is smooth. But when it is activated, when there's an injury and supposed to help in the clotting, to aggregate, to plug up the open part of the blood vessel, then it develops some spiky projections. The spiky projections, sometimes we call this lamellopodia. So these spiky projections help in forming a mesh-like network that plug up the blood vessel where it has been injured, and therefore stop blood from moving out, forming coagulation. However, when you look at the inner part of this platelet, we have the hyalomere comprising of mainly glycoproteins, receptors, and some gelatinous zone with cytoskeleton. So that we refer to as the hilomere. And then at the core of it, we have this central part, which is the granulomere. And that granulomere contains mainly, just from the suggestion, granules. It contains alpha granules containing fibrinogen, dense granules, a lot of lysosomes, and membrane zone that synthesize thromboxane for signaling, so that when you have an injury, this is very important in signaling to bring other cells into the area of injury so that a clot can be formed. And from this central granulomere, we have canaliculi. These canaliculi are very small channels that open to the outside. And it is from these channels that we have the synthesized signaling material moving from the central part to the outside to cause changes in the membrane of the platelet and also to signal other cells to come to the area of injury. So platelets, like their function, is very important in clotting by aggregation, and also there'll be other proteins that are very important, like fibrinogen, which is in the platelet, and then fibrinogen in the blood also is important in that process of clotting. That is what we have as the cells of blood. Let's just review the slides. All this you are seeing here are multi-lobed nucleus. These are the neutrophils. And we said the lobes of the nucleus increase with the age of the neutrophil. Here we have an eosinophil, bisegmented nucleus, and the cytoplasm contains very dense granules, very dense granules. You can see here very dense granules in the cytoplasm. And the bisegmented nucleus is connected by a chromatin strand. These are lymphocytes. These are put at a higher magnification. It's not necessarily the size, but at a higher magnification. And when you look at these cells, they come from the bone marrow. So we are going to look at the bone marrow and the structures within the bone marrow. This is a basophil. Notice slightly larger size than the large size than the erythrocyte. Notice the kidney-shaped nucleus and basophilic cytoplasm. And then we have these granules. These are the platelet fragments that you see between the red blood cells. This is bone marrow. In this bone marrow smear, you can see a whole population of cells. You see cells almost similar to neutrophils, like these ones here, similar to neutrophils, and also similar to eosinophils. C-shaped nucleus. This C-shaped change to become bisegmented, and therefore that differentiate to form a neutrophil. This becomes becomes two lobes. That becomes an eosinophil. So you can see at this stage, the intensity of staining is stronger. So this is the stage where you now differentiate between the normal granules, or also called azurophilic granules, and the specific granules which distinguish the various granulocytes. Changes in the nucleus. The cell initially around nucleus, it becomes an intended nucleus. It becomes C-shaped nucleus. And then some of it might remain as C-shaped, but the granules change, and then it might become now multilobed nucleus. Therefore, we distinguish the various stages in development. On the other hand, we see these other bluish staining cells, which will be in the basophils, but we'll look at those later. So most of what you see here are actually the myeloid series. The myeloid series. A few of these will be the erythroid series. So we look hematopoiesis. What is hematopoiesis? From the word poiesis is formation. Poiesis is formation. Hem comes from blood. So we are talking about formation of blood. Formation of blood. Specifically, we're talking about formation of the blood cells, the formed elements of blood. So we start with a cell that is pluripotential, a hematopoietic stem cell, can differentiate into various types of cells. And then we have differentiation is in various stages. Even before you come to this pluripotential, we have what you call burst-forming units. Burst-forming units of the cells from a hemocytoblast. The hemocytoblast is the main cell, the stem cell that differentiates to different blood cells. So that hemocytoblast, the word blast is an immature cell. Hem is blood. So hemocytoblast is an immature blood-forming cell. So from that hemocytoblast cell, we have burst-forming cells. Those burst-forming cells eventually differentiate to form colony-forming units. We have various colony-forming units. Colony-forming units, for example, for the neutrophilic group. Colony-forming unit for the erythrocyte series. Colony-forming unit for the monocyte series, and so on. These are colony-forming units. Once those colony-forming units form, those are pluripotential. Now the myeloid stem cells, referred as myeloid stem cell. And then from this myeloid stem cell, we have colony-forming units for hemocytoblast, and then we have progenitor stem cells. The progenitor stem cells are progenitor just before the other cells now. Progenitor before myeloid or lymphoid series. Myeloid or lymphoid series. Now, of the myeloid series, we have granulopoiesis, formation of the cells that contain granules. Erythropoiesis, formation of the erythrocytes. Monopoiesis, formation of the monocytes. Megakaryopoiesis, formation of the megakaryocytes. All these are from the myeloid series. A megakaryocyte, monocyte do not have granules. And then we have the lymphoid series. And the lymphoid series, we have the formation of the lymphocytes. Formation of the lymphocytes. So this is what we have as the progenitor cells, from which are differentiating from colony-forming units. So in general, hematopoiesis starts very, very early. By the third week. Second week, we already have in the yolk sac, we have the formation of blood islands in the yolk sac. These blood islands eventually coalesce. Cells differentiate from the mesoderm to form the inner cells, differentiate to form blood cells. The outer cells differentiate to form the endothelial cells. That is in the early stages in the yolk sac. So from that stage, from week 2, why do we need formation of blood in week 2? Because like we said before, the embryo is expanding, is growing, It needs to have nutrients. It's not able to abstract nutrients throughout all the cell. So we initially abstract nutrients from the yolk sac, which is nutrient rich. Then thereafter, we'll abstract from the other parts. So after that, up to the 5th, 7th week, we have in the second month, we have the formation of blood cells now in the liver. So that is the hepatic stage. In the yolk sac, we call that mesoblastic stage. And then in the liver, we refer to that as the hepatic stage. During this stage, you also have some of it in the spleen, and some small lymphocytes have been differentiated. So in the lymph nodes, but most of it is in the liver. So the 5th, 7th week, that is in the second month. Thereafter, as the embryo has developed, has become bigger, we then have changes in terms of formation of blood. Most of it will now shift from the liver to the bone marrow. So the bone marrow of most of the bones in the fetus will be very important in the formation of blood, especially what you refer to as the red bone marrow. So bone marrow forms blood, and this is up to the 20th year. Most of the bones form blood, but after 20 years, the long bones, most of the marrow in the long bones will now be replaced by adipose tissue. So we now have what we refer to as the yellow bone marrow, and most of the bone marrow that is red, that is forming blood, is shifted to the axial skeleton. Axial skeleton. So you know what axial skeleton are? Axial skeleton, the flat bones of the head, the vertebra bones. We have the sternum. We have the ribs. We have the pelvic bone. That is where you have most of the bone marrow forming in the adult, blood being formed in the adult. So within bone marrow, there's continuous stem cells that are very important in generating bone marrow throughout life. So bone marrow then becomes the main hematopoietic organ in the body. So like I've said, the long bones, now the marrow there is invaded by adipose tissue, so it becomes yellow bone marrow. It cannot form blood. However, in very stressful condition, you can have the yellow bone marrow reverting to form blood cells. So within that yellow bone marrow, we still have some stem cells which are capable, and they need some triggers to be able to continue to produce blood cells. So what we are seeing in this slide here is the liver, and you can see the blood cells being formed here and endothelial cells. These are during the formation of the sinusoids within the liver and differentiation of the mesenchyme tissue to form blood. So most of the blood cells are of mesodermal origin, but however, in the yolk sac, that is endoderm, in the endoderm. This one here, at a later stage, you can see already the cells are forming in the liver. This is liver, the fetus over liver, during this hepatic stage. So the 3 stages are mesoblastic stage in the yolk sac, then we have hepatic stage, and then the myeloid stage. Now, when we have the formation of blood, blood is from a hemocytoplasm, we have said that pluripotential cell, but all come from the hemocytoplasm. And then from the hemocytoplasm, we have these 2 series. the lymphoid series for lymphocytes, and the myeloid series for the rest. For the myeloid series, the stem cell, the cell that now differentiates to form all the others is myelocyte, myeloblast. Myeloblast. From a myeloblast, anything with blast at the end is an immature cell. An immature cell, which now matures to form a mature cell that has a site at the end, CYTE at the end. So from this myeloid series, we have the erythrocyte, megakaryocyte, we have the mast cells, we have the granulocytes, we have the monocyte, and then of the granulocytes, we now have the neutrophils, basophils, and eosinophils. This is recognition on the basis of granules. However, the monocyte and the neutrophil come from the same lineage of the myeloid stem cell or the myeloblast. They come from the same lineage as the myeloblast. So we have a colony forming unit, neutrophilic, colony forming unit, myelocyte. So this is how the cells of blood, formed elements of blood, are coming from. This is just to the differentiation of the cell from the hemocytoblast or stem cell. Then we have the common myeloid progenitor cell. We have the megakaryocyte, which forms the thrombocyte or platelets, megakaryocyte, and then the erythrocyte forming red blood cell, mast cells, which are in the tissues, mainly playing a role in inflammation. Then we have the myeloblast that will form the granules here, and the agranular, the monocyte. So all this from the myeloid stem cell. Then we have now the lymphoid stem cell. I said the natural killer cell is larger, slightly different, but there are T cells, natural killer cells, and then small lymphocyte, the T lymphocytes and the B lymphocytes. So this is how they originate. So let's look at each of those and see. This is just a summary of the same thing we have talked about. Progranulocyte, eventually forming granules, and then the agranulocytes not forming granules. But you note the monocyte, which is a granulocyte, will be coming from the same, like that of the basophil, all from the myeloid series. For the red blood cell, we have hemocytoblast, like we said before, which is the myeloid stem cell eventually. Hemocytoblast, like we said, splits into myeloid stem cell and lymphoid stem cells. So the myeloid stem cell, from the myeloid stem cell, we have proerythroblast. Then the proerythroblast, the organelles are becoming synthesized. We have formation of the hemoglobin, the proteins being formed, hemoglobin. So we have a proerythroblast. As a result of increase in the hemoglobin formation, we have now the cytoplasm starts staining basophilic. In that case, we now have basophilic erythroblast. Why are we calling it erythroblast? Because it's yet immature. It's not a mature cell. Then we have this stage where we have an increase in the number of organelles, increase in the proteins within the cytoplasm, increase in the production of hemoglobin. So the cell now takes different stains, it becomes metachromatic. It takes different stains with different colors. So we have polychromatophilic affinity for various different dyes. So we have a polychromatophilic erythroblast, which differentiates to form orthochromatophilic erythroblast. Now, at this stage of orthochromatophilic erythroblast, we have been talking about changes within the cytoplasm, but we also have the changes in the nucleus. In the myeloid stem cell and the proerythroblast, we have a very, very large nucleus, very large nucleus, almost occupying the entire cytoplasm. The nucleus starts shrinking. It undergoes a process we call pyknosis. So during that process of pyknosis, in the polychromatophilic erythroblast, the nucleus becomes very small, migrates towards the cell membrane. And then at the stage of the chromatophilic erythroblast, the nucleus is extruded from the cell at the orthochromatophilic erythroblast. This stage is also called normoblast in some books. So the nucleus is extruded. So we have an extruded nucleus. Here, remember, there's no nucleus. It has been extruded. We have the reticulocyte, which is still a round cell. There's a lot of organelles. We still have some slight protein synthesis in the reticulocyte stage, but the reticulocyte now undergoes morphological changes. It undergoes morphological changes once the nucleus has been extruded. It now becomes biconcave discoidal in shape. So the reticulocyte is pushed from the bone marrow into blood. So some of the 2%, 2 to 5% reticulocytes will normally be in normal blood smear circulation, about 2 to 5%. But most of it will be still differentiating within the bone marrow. Then that morphological change now become mature red blood cell, means there is changes in the shape of the red blood cell. This is, you can see a very large nucleus in the proerythroblast. The nucleus becomes small. Let's use this particular slides here. Very large nucleus. The nucleus becomes small. The cell also becomes small in size. The nucleus undergoes pyknosis. That's what they're calling pyknosis. It moves at the polychromatophilic stage towards the cell membrane, and eventually is extruded outside by the process of exocytosis. The nucleus is extruded. So that cell, which has an extruded nucleus, but still very rich in organelles, still synthesizing proteins, hemoglobin, that cell is referred to as a reticulocyte. The reticulocyte then changes shape and becomes biconcave discoidal. So when you look at this, you can see this is a concave surface. And if you flip it over, there's also another concave surface. Concave on both sides, but it's a disc-shaped structure. And biconcave discoidal. We have more cytoplasm on the periphery of the cell. So the peripheral part of the cell has more cytoplasm. So it is deep staining, more hemoglobin. So when you look at it on a flat after staining, the central part looks hollow. You can see the light stained central part because there is less cytoplasm. This change of shape to a biconcave discoidal, and the maintenance of this biconcave discoidal by a protein called spectrin. Spectrin is a protein that maintains the shape of the red blood cell as biconcave discoidal. This impacts flexibility to the cell so that it can be able to move through very narrow capillaries. So the red blood cell can pass through very narrow capillaries because it's very flexible. It can be able to move very easily. Removal of the nucleus is to create, and changes to biconcave shape, biconcave discoidal shape, is to create more surface area for the absorption of oxygen, primarily oxygen. You create more surface area. That's why we have this biconcave discoidal, and that is the erythrocyte. So imagine a cell as big as this, as the hemocytoblast, eventually undergoing mitosis, forms the myeloid proerythroblast, and then from proerythroblast, the nucleus becomes smaller, and then eventually is extruded, and the cell has no nucleus. So a red blood cell has no nucleus. There's no nucleus, biconcave in shape, the biconcave shape maintained by spectrin. The biconcave shape, removal of the nucleus, is to create more surface area for the absorption of oxygen. That is a red blood cell. And usually in a blood smear, the red blood cells appear in stacks. They appear in stacks. And those stacks, we refer to them as rouleau formation. Rouleau formation. And also, as they move in the capillaries, they don't just swim through, they actually roll on the endocalyx of a capillary. They roll on the endocalyx of the capillary because of the cell adhesion molecules, the integrins within the cell membrane. And they're able to pass through also the capillaries because of the cell adhesion molecules within their membrane. So that is the red blood cell, primarily transport of oxygen. Anatomically, increase in surface area to be able to absorb more oxygen by creation, one, removal of the nucleus, and also creation of a biconcave disc-shaped cell. Once the nucleus is removed, we do not have any more synthesis of proteins. However, in the reticular stage, we still have some synthesis of proteins. So what happens when you have very few blood cells being formed? When you have red blood cells being formed, very few of them, then you have anemia. Anemia. Vitamin B12 is very, very important in a process of erythropoiesis. Vitamin B12 is very important in the process of erythropoiesis. And there's a hormone called erythropoietin, produced by the endothelial cells of the kidney. Endothelial cells of the kidney. Erythropoietin is a hormone that stimulates erythropoiesis. Usually under low oxygen tension, that is hypoxia, then you have stimulation of production of erythropoietin, which then stimulate the bone marrow to produce more red blood cells. The lifespan of a red blood cell is normally 120 days. 120 days. So we have anemia. When you have problems in the digestive tract, we have a cell called parietal cell in the stomach, parietal cell, that apart from producing hydrochloric acid in the stomach, also produce gastric intrinsic factor. The gastric intrinsic factor is very important in the absorption of vitamin B12. Vitamin B12 in the digestive tract. So if there's a problem with the production of gastric intrinsic factor from the stomach, then you have absorption of vitamin B12 becomes a problem. Then we have more cells are being produced that are immature in circulation because the body senses that there is less oxygen So it needs more red blood cells in circulation, and therefore the bone marrow produce more and more cells and push more immature cells into circulation. So that is what we refer to as megaloblastic anemia. Megaloblastic anemia. Very large immature cells that are not capable of absorbing oxygen are being produced. Megaloblastic anemia because of vitamin B12 deficiency caused by lack of gastric intrinsic factor because of an effect on the parietal cells in the stomach. The other cells also, we can have other cells being produced more, but we look at those when we're looking at bone marrow. So let's look, that's about blood. Let me look if there's any question on blood. Inflammation. The inflammation, which is swelling as a result of any injury, has two roles. One, there is increase in permeability of the blood vessels. This increase in permeability is produced by histamine. Histamine can produce increased permeability of the blood vessels, and therefore, as a result of the increased permeability, we can have more plasma getting out into the perivascular space. That's one. Two, we also have production, we have more vasodilation. There's also more vasodilation, and this vasodilation increase diameter of the blood vessels, the capillaries, will also increase blood flow to the area of injury. As a result, we also have recruitment of the cells. So recruitment of other cells like neutrophils, basophils, to come, neutrophils specifically to mob up destroyed substance, to phagocytize any dead tissue or bacteria. Normally it will attempt to phagocytize, and if it's too much, that's why we have pus. And neutrophil is also referred to as a pus cell. Pus formation is because the neutrophil is trying to fight to remove the dead cells and dead bacteria and anything that is within this area of injury. So the swelling is important because of more recruitment of cells and also more exudate of plasma into the extravascular space. And it might also be because of edema. Edema being lymphatics cannot cope. The lymphatics cannot cope with this efflux of blood. Efflux of blood. Can you mute your microphone, please? Milton, mute your microphone. Let's look at bone marrow. We have said bone marrow is a hematopoietic stem cell. It's a hematopoietic organ. It's universal hematopoietic organ because it's producing all the formed elements of blood. We normally distinguish red bone marrow, which is the site for hematopoiesis, and yellow bone marrow. Red bone marrow and yellow bone marrow. The marrow spaces, that is the areas where you have the actual formation of blood cells, are separated by bone tissue. We have bone spicules separating the bone into these spaces. This mainly in the spongy bone, the spongy part of the bone. So those areas of the spongy part of the bone, the inner part of that bone is covered by an endosteum. Remember, the outer part is periosteum, the inner part is endosteum. Within these endosteal covered areas, This is where we now have the hematopoietic structures, the red or yellow bone marrow. The marrow is the spaces, not the whole bone, but the spaces. Now, I've said red bone marrow is areas for formation of red blood cells, active production of formed elements of blood. The yellow bone marrow is where we have replacement of the active cells by adipose tissue, white fat. This white fat, or the adipocyte, absorbed some of the dye-like structures. That's why it's yellow. Yellow bone marrow revert to red bone marrow, I've said that before, under excessive blood loss. Excessive blood loss. And also, what does excessive blood loss mean? You have lost a lot of blood. It means the body recognizes that there is less blood in circulation, less hemoglobin transferring erythrocytes in circulation, and therefore, as a result of that, the sensors, like I've said just before, erythropoietin is stimulated, erythropoiesis is stimulated to produce more cells. So where there's been a lot of loss of blood and it's not immediately reverted, then some of those, the red bone marrow cannot cope with the demand for more blood cells. And therefore, we have some of the stem cells in the yellow bone marrow being activated to produce more blood. In bone marrow, just like any other organ, reticular fibers form the supportive framework. I think we talked about it when we were talking about tissues, if you remember. So reticular fibers are the main supportive framework of bone. Reticular fibers are synthesized by reticular cells or also fibrocytes. Bone marrow is devoid of lymphatics. There's no lymphatic in bone marrow. Now, in the bone marrow, if we take in a section, we are able to identify 3 parts. We are able to identify the supportive structures that support the cells, which we refer to as the stroma. So the stroma, comprising of the reticular fibers, the astrocytes, the fibroblasts, fibrocytes, and adipocytes. So that forms the stroma, separating these other parts. Then we have the vascular compartment. The vascular compartment mainly containing of sinuses, blood sinuses. This is where we have the sinusoidal capillaries. If you remember when we talked about sinusoidal capillaries, we talked about sinusoidal capillaries being very wide diameter. The cells leave very large gaps between the cells, intercellular spaces. In the very thin ends of the endothelial cells, we have very large openings. This is to facilitate passage of cells. The mature cells can pass from the hematopoietic compartment to the blood sinuses, where they are then transported to the rest of the body. The other compartment is the hematopoietic compartment. And just from the term hematopoietic, poietic, this is formation. Poietic means formation of blood cells. In the hematopoietic compartment, this is where we have differentiating cells of the myeloid and the lymphoid series together with the together with the stem cells. Together with the stem cells. So these ones are differentiating cells. We have them in the hematopoietic compartment. So we should be able to distinguish these three compartments, specifically the vascular compartment and hematopoietic compartment. Like I've said, bone marrow is a universal hematopoietic organ. So the formed cells, the mature cells, move into the vascular compartment and then transported throughout the body or to the other cells for colonization. Now, in the histology, if you examine a bone marrow and you examine a blood smear, what's the difference? In the red bone marrow, what will be the main cells that you are going to see? We have said red bone marrow produce cells. So we should be able to see cells of the myeloid series. We should be able to see cells of the lymphoid series. And we should also be able to see the fibroblasts. And also the adipocytes. Sinusoidal capillaries, I think I've talked about that. Functions of sinusoidal capillaries in terms of allowing mature cells to move. It allows mature cells to move into the sinusoidal capillaries, but does not allow immature cells. Selectively does not allow immature cells unless under very critical conditions. So when we have a lot of immature cells, what does that mean? It would mean either that the sinusoids have been compromised or there is too much production, too much demand that the sinusoids now allow these immature cells into circulation. We have talked about megakaryocytes, a very, very large cell, very large nucleus. And I said we have karyokinesis without cytokinesis. The nucleus chromosomes divide, they double, but the cell does not divide. The cell does not divide. So what are some of the adaptations of the bone marrow to be able to perform this function? What happens? Just look at this tutorial problem. A patient moves from a coastal city to a high altitude region. Over time, their bone marrow adapts. A student comes from Mombasa, or a lady comes from Mombasa, that is sea level, comes to either Mount Kenya or even here in Eldoret, very high altitude, and after some time is able to cope. What happens at high altitude? What happens at the high altitude? What happens at low altitude? What happens to the pressure changes? What adaptations do you think should be there at high altitude? Why do people, when you suddenly go to a high altitude, start breathing very fast? Because the air oxygen concentration is low. Air pressure is low. So you breathe fast to be able to get in as much oxygen as possible. So what changes occur in the red bone marrow, the yellow bone marrow ratio, and why? what did we say happens to yellow bone marrow? This person is moving from a low altitude to a higher altitude, has to adapt. But immediately there's a lot of stress. There's low oxygen concentration. The pressure is very low. So what happens? There's demand for more and more of hemoglobin, more red blood cells. And therefore, you now have the yellow bone marrow reverting to produce now more blood cells, more red blood cells. You have yellow bone marrow reverting to produce more blood cells. And therefore, the ratio of the red initially at the coast, the red will be higher than the yellow. But now the ratio is changing so that you now have the red being close to the yellow, similar to 3 to 1. Which hormones drive this adaptation and why? You move from the coast to the high altitude. Hypoxia is the key. The key word there is hypoxia. So you have hypoxia, low oxygen. Hypoxia stimulates production of erythropoietin. Hypoxia is monitored by the tissues in the carotid body and also the aortic body. So those monitor oxygen concentration, carotid body and aortic body. The information is then sent to the kidney, the endothelial lining of the kidney. The endothelial cells in the kidney then produce the hormone erythropoietin. Erythropoietin is transported to the bone marrow and stimulates the process of erythropoiesis. The process of erythropoiesis. What do you think would happen immediately after that person comes? There'll be more throwback of reticulocytes. The number of reticulocytes in circulation will increase, but after time, eventually that stabilizes. How would the bone marrows differ? Immediately before and after adaptation in terms of cells. So here, purely we are talking about oxygen demand in terms of bone marrow. If I take a yellow bone marrow and take a section, I will see some increase in islands of red, where we now have yellow bone marrow producing the erythrocytes. This is a section of the bone marrow. In this section of the bone marrow, this part here is the vascular compartment, lined by an endothelium. So this is a vascular compartment, the sinuses. These are the red blood cells and some of the cells that are in circulation. Neutrophils, basophils, eosinophils, and so on. This one here is the hematopoietic compartment. Hematopoietic compartment. We have cells that are differentiating. Once they differentiate, they then pass through the openings, apertures within the vascular compartment into the vascular compartment and are transported and are now transported to the other tissues, to the other parts of the body. You can see these ones are already differentiated. They're almost neutrophils. You can see the mild lobed nucleus, neutrophils. This one here, a kidney-shaped nucleus, but the cytoplasm is staining red. So most likely differentiating to form, going to differentiate to form an eosinophil. After that, it will now move into circulation. Amongst these cells, we have adipose tissue, adipocytes. This is a large capillary. We have adipocytes. So the adipocytes will eventually take over most of this yellow bone marrow. When we look at yellow and the red, we'll see mainly adipocytes in the yellow bone marrow. So that is the difference between the bone marrow, the compartments of the bone marrow. Apart from this, we have, of course, supporting these cells, we have reticular fibers and reticular cells. Reticular fibers and reticular cells. This is a section of a bone marrow, the same. This is adipose tissue. This is a red bone marrow. This is adipose tissue. These are adipocytes. Adipocyte. These are megakaryocyte. Megakaryocyte. And this part here, you can see a lot of these cells here. These are the cells, these are hematopoietic compartment. Hematopoietic compartment. With a lot of differentiating cells. Note this other cell, which is a band cell. Bone marrow. Bone marrow. This is a myeloid, the developing cell is a myelocyte. These are metamyelocytes, so we'll talk about them later. It's a metamyelocyte. These cells are differentiating. This one here, looking at it, you can see a very small nucleus. This one is of the erythroid series, the polychromatophilic erythroblast, that eventually is going to differentiate to form an erythrocyte. Now, for the granulopoiesis, we said granulopoiesis comes from the myeloid series. From the myeloid series. And we have several stages. We have a myeloblast, if you remember, an undifferentiated cell. An undifferentiated cell, very large nucleus and so on. Differentiating from the hematopoietic stem cell. Hematopoietic stem cell differentiates to form a myeloblast, which then differentiates to form a promyelocyte. Promyelocyte. This stage here is a promyelocyte. Notice the changes in the shape of the nucleus. Notice the changes in the staining characteristics of the cytoplasm. And then the promyelocyte differentiate to form myelocyte. This stage of the myelocyte is where we have recognition of the specific granules. The promyelocyte, notice the nucleus changes. Intended, initially it was round, becomes intended. It becomes C-shaped. We refer to this also as a band cell. Band, B-A-N-D, band cell. But at this stage, we have granules differentiating. Notice this one here is pale staining. This one is deeper stained granules. So the myelocyte stage is where we can now differentiate the myelocyte stage. So this is a myelocyte stage, sorry. The myelocyte stage is now where we can differentiate the basophils, eosinophils, and neutrophils at the myelocyte stage. So we should now use those terms: neutrophilic myelocyte, basophilic myelocyte, eosinophilic myelocyte. Then after that myelocyte stage, we have a metamyelocyte. This is the stage of the metamyelocyte, sorry. So stubs also forming what you call stub cell. C-shaped nucleus, and from this metamyelocyte, the nucleus completely transforms and becomes either a neutrophil with several lobes, an eosinophil, or a basophil. Basophil is not in this, but we have an eosinophil here with very large coarse granules. And then a mature cell whose structure we have described. Megakaryocyte, I think we've talked about megakaryocyte just to show the picture. The whole of this is a megakaryocyte. This is a cell that has undergone what you call endomitosis. Endomitosis. And in endomitosis, the nucleus divides, the DNA increases, sometimes up to 2, times 46. DNA increases without an increase in up to 96. DNA increase without increase in the division of the cell. The cell does not divide, but the nucleus divides. So we refer to that now as endomitosis. And then the process of this megakaryocyte, the process of this megakaryocyte here projects into the vascular compartment, which we had seen earlier. It projects into the vascular compartment and pinches off. It pinches off now as platelets. This is just a repeat of what we had seen before to show red blood cells. You can identify red blood cells here. You can identify the differentiation of the neutrophil. This here is a myelocyte stage. You can identify the metamyelocyte stage of the neutrophilic metamyelocyte stage. You can identify the development of the red blood cell up to this stage where the nucleus is now very small and eventually gets extruded. And then you can note the rule of formation of the erythrocytes, the stacking up of the erythrocytes to form rule of formation. You can see this is a basophilic metamyelocyte, basophilic. Look at the cytoplasm, is basic and the nucleus is kidney shaped. This is just a bone marrow again, bone marrow smear. Bone marrow smear, just to show the extrusion of the nucleus, pyknosis of the nucleus, and subsequent extrusion forming a reticular cell. Notice reticular cell, the staining characteristic of reticular cell as compared to the erythrocyte. The shape of the reticular cell as compared to the reticulocyte, where the reticulocyte is biconcave discoidal, the reticular cell is still spheroidal. And here the neutrophil, note the bar body of the neutrophil. And here the monocyte. That is about blood and well-known questions. Any question up to there? Let's deal with the bone marrow to bone marrow. Fine. In the absence of a question, I'd like us to just deal with the primary organs, and then we'll go to secondary organs in our next class. I'm sorry, I think we'll stop there for today. We are going to start on the lymphoid organs per se, the lymphoid organs in our next class. I now invite any questions. You can put them in the chat or ask directly. Marcus, can you ask your question? Nyongesa?
It was answered.
Okay. Anybody else with any question? Fine. So we'll stop there for today. I'd like us to have another online class to finish